I'm so glad you're here. My name is Amy Lehman. I'm a statistician in the Center for Biostatistics in the Department of Biomedical Informatics. And the title of my talk today is Statistics and Research, Thoughts from a Biostatistician, which I just realized is not the official title of the talk. I just remember looking at the thing. The official title of talk is actually the role of statisticians in research. So the goal of this talk today is not to really teach you statistics, because we can't talk about grant writing in an hour. We definitely can't learn statistics in an hour. But my goal is to try to give you a brief overview of the current state of statistics and research, talk about how statisticians fit into research, and then hopefully give you some very useful tips that will be relevant for you as you move forward in your research careers. That's my goal. We'll see if I achieve that. Before I start, though, I kind of want to acknowledge, reality, and because I'm very aware that there are lots of things you can do without a statistician, right? I mean, we all know this. One thing you can absolutely do without a statistician is you don't need a statistician to find out information about statistics, right? So for those of you beyond just simple coursework, there is so much information available to you online. There are courses, there are tutorials, there are websites, forums. There's so many ways for you to access information about statistics on your own or in groups. And you can definitely conduct all of your analyses without a statistician, correct? I mean, technology has given us so many different types of software. There are many choices of things that you can use to kind of easily analyze your data. So you probably have your favorites, everything ranging from Excel to R, SPSS, Prism, I know, if you're familiar with that, is a popular choice, Stata, et cetera. It's never really been easier for you to do even complex analyses where before you had to have some kind of really sophisticated programming knowledge. And it's absolutely possible to get published and be successful in that way without a statistician as well. Either if you've not done that yourself, I bet you probably have colleagues or even mentors or superiors who have published and gotten grant funding without a statistician. So saying that, knowing all that, I always wonder when I give this talk if people think me as a statistician saying you need to work with a statistician is kind of like this pushy salesman trying to sell you a product you really don't need, correct? And I want to say that I understand. I totally get where you're coming from, because let's face it, we are in a DIY culture, correct? With information, there's no really need sometimes to kind of rely on experts. When I need to learn how to do something, my first reaction is not to set up an appointment with an expert, but I'll go online and see if I can figure it out myself, right? And I think there's many good things about that. But, the caveat, what's the worry about this? The problem is that with the DIY approach is that sometimes having a superficial knowledge of a subject may lead to kind of a suboptimal result, correct? DIY doesn't always work the way you want it to. And I would also argue that there are certain fields or areas where we should not have a DIY mentality, okay? This is an actual question. I did not make this up. If you actually go online, which is an amazing place, you will find this question about becoming a self-taught surgeon, okay? Now, that's nothing, I'm sure the person who asked this question is very motivated and intelligent, but none of us would probably say self-taught surgery is a best practice or a way to go. I mean, that doesn't mean that every single surgery would be unsuccessful, but we all know that that is dangerous, okay? I'm gonna argue that self-taught statistics is also a dangerous practice to have as well. And the reason why I'm saying that is because you as researchers are under a tremendous amount of pressure to publish, to get your research out there. And we are publishing an extraordinary amount. I mean, I don't know exactly how many scientific articles there are out there, but there are a lot. And the system that we're under currently rewards those of us that publish a lot, right? One of your metrics probably for advancement in your field is how much do you publish, right? Have you had 70 articles, you'd probably be super happy, because that would just be an amazing metric. The problem is that we are publishing so much that I'm concerned that we are focused on quantity over quality. Sure, we're getting papers out, but are they actually reflecting real, reproducible results, okay? And so there is a growing awareness of a real problem in research today, and that is the problem of irreproducible study results. And just you might notice that I'm kind of flipping through a lot of these articles and things really quickly. I'm sorry because of time, but just so you know, I do have a handout with references to all these articles, which is available to you afterwards, so you can relax if you want to kind of look at these at your leisure, and all that free time you obviously have afterwards, feel free to go. So you can just kind of relax and enjoy the stats talk. All right, but it's made the news lately, and there is a growing awareness of a real problem having findings that can neither be validated nor replicated, and just this real concern that the majority of positive findings we have today will turn out to be false positives. In other words, scientific dead ends that waste time, money, and resources. So here's a fairly recent example. This was an interesting study. This was published in the journal Cortex in 2015. Its sole purpose was to replicate the results of 5 previous studies that looked at structural brain behavior correlations, or as I understand it, relationships between the brain's structure with behavior and cognition. So what did these researchers find? Here's a summary of the results. The 5 studies we attempted to replicate contained a total of 17 structural brain behavior correlations. The results from our confirmatory analyses showed that we were unable to successfully replicate any of those 17 correlations, okay? Now I want to mention, just because this one study did not replicate those results does not automatically make those 5 studies false or invalid. But that does raise concerns in my mind about the reliability of any study that we read in the literature, because we are trained to believe that studies that are published in reputable peer-reviewed journals as accurate, right, or indicative of the truth. But the problem is that not even well-regarded studies are immune from this problem. Here's a second example. This study was published in the Journal of Clinical Endocrinology and Metabolism in 2008. This was an NHLBI-funded study that found a link between polycystic ovary syndrome in postmenopausal women and cardiovascular disease. From 2008 to 2015, this study was cited over 300 times. I believe it was like 321 times by Google. But what happened in 2015? As it turns out, the authors had to withdraw their study due to their inability to replicate the original results, okay? And here's a final example. This just happened. How many of you have heard the news about the Mediterranean diet study retraction? Anyway, this was a big study. So there was a large randomized clinical trial published in the New England Journal of Medicine showing that participants given the Mediterranean diet experienced cardiovascular benefits. But what happened? After some investigation, they realized that there were issues with the way they randomized the patients. The authors were forced to very publicly retract their study and republish a revised analysis in which the link between the Mediterranean diet and cardiovascular disease was much softened, okay? So again, I don't have a lot of time with you today to talk about this. These are just 3 examples, but please believe me that these are not isolated incidents. You know, the problem with irreproducible study results is well known and unfortunately pervasive in today's research. In 2016, the journal Nature sent a survey out to over 1,500 scientists asking them about this problem of reproducibility. And what they found was maybe not too surprising, but not very happy. It said that more than 70% of researchers have tried and failed to reproduce another scientist's experiments, and more than half failed to even reproduce their own experiments. Okay, so this is definitely a problem. But what I personally found interesting is they asked those same researchers what could be done to boost reproducibility. And what do you think they said? The number one factor they thought that could increase reproducibility was a better understanding of statistics. Now again, I mean, this is not a scientific study. This was just a very small, convenient sample of some scientists. But the results here are consistent with what's being written about statistics in research. This is a great article in 2015 about the misuse and abuse of statistics in research. And again, all these references I have available for you in a handout if you'd like to look at these later. But here's a great quote that I think is really relevant to research today. It says, the computers and statistical software packages have increased the complexity with which data can be analyzed, and consequently, the use of statistics in medical research has also increased. Unfortunately, though the types of errors may have changed, the frequency of statistical misuse has not. These errors are primarily due to inadequate knowledge and researchers not seeking support from statisticians. I mean, how common is that you read about some cool new method in a paper and think, I want to do that to my own research, maybe without completely understanding the theory behind it or if it really works. Technology makes that very easy to do. Maybe that might not be the best thing to do, though. Here's a final article I want to give to you, very recently written just last year about the reproducibility crisis. And here's a nice quote from there. Without qualified statistical experts to guide them, researchers will continue to encounter big problems. Statistics is demanding in the same way as flying a plane, but many scientists only have the equivalent of a driver's license. As a result, they're crashing into the side of a mountain on a regular basis. Okay. Now again, I'm a statistician, so obvious bias right here. But I hope that these small articles I've given you have given you a flavor for the problem of irreproducible research. This is a problem. And I hope you believe me when I say that bad statistics are a major contributing factor to irreproducible research. And this is why I truly believe that good statisticians are essential members of the research team. Now, later this afternoon, my colleague Guy Brock will be talking about reproducibility and rigor in research. So I'm kind of just giving you the very fluffy introduction. I would really hope that you would stick around because I think that will be a very, very useful talk for you about that. Okay, so we've all agreed now that statisticians are useful, right? I have totally convinced you. Just nod, say yes, so we can, yes, okay. So, all right, I'm a consulting statistician. In January, I will have been here for 18 years working with researchers. And so that's kind of a long time when I start thinking about that. So I started to think in working on this talk and talking with my colleagues, what are, based on our experiences, the most useful things I could tell a researcher from a statistician's perspective to be successful in research? If I could give you my top tips for success in research, what would they be? Okay? And so I have developed for you my top 3 tips for successful research, okay? And here's tip number 1. I'm sure you cannot wait. But this is tip number 1 I have. For successful research, please talk to a statistician before the data are collected. Okay, so to understand that, let's just kind of briefly review a generic research timeline. I'm sure you're all really familiar with this. But most research kind of starts off with a kind of a period of design and planning, right? So moving from left to right, we generate a hypothesis, we write up a concept, get a proposal together, a grant, for example. We then submit that for either internally, externally, for funding or approval. Hopefully after not too much time, you get approved, you go off and you run your research, you collect your data, you run your experiments, you finish data collection, and then you do some sort of a statistical analysis and you disseminate your findings, poster, presentation, manuscript, etc. Oftentimes, the first time a statistician is involved is at the end here in the statistical analysis section. And that makes sense, I guess, because that's the actual analysis, right? You need those p-values, so you bring it to the statistician. But as you can see by the very sad face, that we don't like that, because when you come to us at the end, it's way too late to fix anything that could have gone wrong in the beginning of the experiment, okay? So really, if you're thinking about the research timeline, we want to actually get involved at the very beginning. I think sometimes researchers feel that before they come to a statistician, they need to have a completely worked out research plan, everything kind of put together, and they just need a little paragraph for us to plug in. Absolutely not true. We want to help you as you're even working out your concept, working out your aims. You don't need to have that done. And when we say come to us before the data are collected, we actually also don't mean coming to us 2 days before you have to submit your grant. So Dr. Hall, who gave those great presentations, said he works really well, with like 2 days of, working overtime. Well, and he said maybe some people wouldn't like that. Well, as a statistician, that kind of, we don't like that because a lot of times that doesn't give us enough time as well. And to understand that, why kind of rushing at the end or coming to us late is not optimal, I have an example that I want to kind of talk to you about. Let's switch gears a little bit and talk about food, okay? So I have a question to ask you guys. How many of you guys like to eat ham? Raise your hand. This is a no judgment zone, so if you don't like to eat ham, fine. How many of you guys like to eat bananas? All right. I mean, bananas aren't my favorite fruit, but not bad. Now, for those of you who said yes to both ham, how many of you said yes to ham and bananas? Okay, well then you're going to be super excited about what I'm about to show you, because I found a recipe for a dish that combines the two things that you say you like into one wonderful dish. It's called the ham banana casserole. This is a real recipe, a real honest-to-goodness recipe that was published in 1972 in this McCall's Casserole Cookbook. Okay? You might be wondering what this is like. Well, I'm going to show you what it's like. Okay? So let me show you how you make a ham banana casserole. Okay? And just the last time I talked about this, someone actually thought this was me. I have never actually made this. These pictures come from this amazing website, midcenturymenu.com, where they reproduce old recipes. Anyway, to make a ham banana casserole, you first take lunch meat ham and then smother mustard over it. Okay, nice even mustard, all pretty. You then take bananas and you roll the ham and the bananas in the ham. Okay, you place those lovely rolls in a casserole dish, and then you kind of brush the exposed ends with butter, because butter makes everything better. And then you take all of those rolls and you smother it with cheese sauce. Okay? It sounds amazing, doesn't it? I mean, I'm getting you ready for lunch here. This is going to be good. All right, the people in Mid-Century Menu, they meticulously follow the recipe to the letter. So everything in here was done exactly according to the recipe. But the problem is when they threw it in the oven, this is what it came out like. And then the lady fed it to her husband, and this was the look on his face. Okay? At this point, you're probably regretting not going over to the management, to the other room, correct, where you could have a very informative seminar. Why am I talking about this? I mean, I'm not that insane. Why am I talking about this disgusting recipe? Because I think there could be parallels between food and research. So the reason why this ham banana casserole recipe failed is not because they did not follow the protocol correctly. It's because the recipe itself was flawed. Same thing in research. If your experimental design is flawed, it doesn't matter if everything else that follows after that is done perfectly. If you use the correct statistical test for your data, your outcome is still going to be the ham banana casserole of research, okay? And so the reason why we want to talk to you beforehand is we want to help you avoid design flaws that will doom your experiment. I'm just giving you a quick list. This is not comprehensive, but I personally have seen all of these in my years of working. Bad randomization. That is what actually one of the reasons that the Mediterranean diet study failed. Inappropriate control group, or sometimes forgetting a control group. Collecting key variables in a way that makes analysis difficult, or forgetting important information. I mean, we all know that after you collect data, it is actually impossible, really, to go back and gather information that you forgot, whether it's chart review or actual prospective studies. Inadequate sample size. This is a huge one. And this is probably the one that researchers are the most aware about. The question is, we're always worried about going to all this effort only to find out that we did not have enough to detect an effect. So one thing we can do is actually tell you if your project's going to be feasible. If you need 8,000 patients to detect an effect, you might not even want to do that. Problems with data capture. So not just the design, but how you will be collecting and storing that data as well. All of these things are issues that we as statisticians can help you with as you're working on your experiment. But the number one thing I think a statistician can help you do is focus your research. This is the number one contribution I have. I'm going to switch over to the keyboard because I want to make sure I have control over this. And I want to show you a brief movie clip actually now from a 1995 movie called Sabrina. Has anyone heard of this movie at all? Yay, Tadovik. Okay. I'm not surprised if most of you haven't heard of it. It's an older movie, and frankly, it wasn't that memorable. It's actually a remake of a much more famous version with Humphrey Bogart and Audrey Hepburn, for those of you who are into movies. But you don't need to know that. Not a very good movie. But there's a scene in this movie that I absolutely love and I think is applicable to research. So let me set this up for you. I hope this works. In this scene, a photographer is talking with her client about pictures, about taking photos, okay? And how do you choose what photos to take? So let me see if I can get this to work for you.
Do you want a view from the house?
Sure.
Which one?
All of them.
More isn't always better, Linus. Sometimes it's just more. Pick one.
Okay. So more isn't always better. Sometimes it's just more. So have you ever had that with your phone? You just take like 8 million pictures and you wind up throwing away 99% of them. Same thing happens in research. I know when you're designing your project, you sometimes want to make it as broad as possible, right? Because you're afraid of not catching something important. You want to collect every possible variable there is because you want to make sure that you're catching everything. But that sometimes is not the way to go. In my experience, the most successful research is not the one where you actually capture everything, but sometimes the most targeted. Feasibility-wise, and we've seen that, if you try to collect 500 variables because you're afraid of missing just one, you might not even be able to kind of finish accrual. So a good statistician can help you find that balance between finding enough information, and not going overboard. All right, so keep this in mind. If you're ever tempted to go it alone, remember what Vanilla Ice said.
All right, stop, collaborate, and listen.
Okay. You love my super current references, don't you? I bet a lot of you are like, I don't even know who that is. But anyway, stop, collaborate, and listen. Every time you're tempted to go it alone, and I understand working with a statistician is a little more difficult, it's easier to do it yourself, stop, collaborate, and listen. So that's my number one advice for you. Okay, work with a statistician, come to us before the data are collected. My number two top advice for you as success in research is to please be careful about p-values. Please, please, please. I wonder how many of you know what a p-value is. I'm not going to ask you to define it for me, but you might not be able to define it, but you kind of have a sense of what it means. You've seen them before, you've computed them. Okay. If you have not seen them before, I guarantee you in research you will, because p-values are sort of the currency, currently the currency by which we measure research significance, correct? If you're going to make a scientific claim and submit it to a journal, most likely that journal will request to see a p-value to back up your results, correct? So why am I saying be careful about this? So to do that, let me review just the typical scientific framework we have. Again, if you've taken a stats course, this is probably what you've been taught before. So hopefully this will be a review for you. So typically when you're asking a scientific question, you do something, you construct what's called a null and alternative hypothesis. So recall that the null hypothesis is usually considered the status quo, what you assumed to be true unless you have strong evidence to reject. So a lot of times it's the no difference. You can think of it as the no difference hypothesis. Then you have the alternative hypothesis, which is actually what you want to prove, right? But you will not kind of conclude the alternative unless you have strong evidence in favor. So for example, if you have a new experimental drug, your null hypothesis might be there's no difference between my experimental drug and placebo, where the alternative might be there's going to be a big difference in outcome between the two. So you have these two competing hypotheses. You then design and run an experiment. You apply a hopefully appropriate statistical test, and you get something that's called a p-value. What is the p-value? The definition is it's the probability of observing your results or something even more extreme, assuming the null hypothesis. Okay, so the idea then that is if the p-value, if the probability of observing your results, assuming the null hypothesis is true, is so small that it's unlikely, we will reject the null hypothesis. Otherwise, we fail to reject. So in other words, if we assume no difference with this new drug and placebo, and then we run a sample of 100 patients and we see that the people on the new drug are 50 points higher than those with placebo, how likely is that there really was no effect of the drugs? Okay, it's a probability. And normally, typically we use 0.05, so 5% probability, okay? And so that's pretty much what we do, and then kind of repeat as needed for every experiment. Does this seem vaguely familiar to you guys, or very familiar, or at least vaguely familiar to you guys? All right. This is the standard scientific framework that we are taught in statistics school. I mean, it is so ubiquitous that it even makes it onto joke t-shirts that only statisticians find remotely amusing. I would totally buy that t-shirt, so that totally says everything you need to know about me. All right, what is wrong with this then? Why am I saying be concerned? And to really appreciate that, I want to give you a very, very brief history into the p-value for those of you who may not be familiar with it, because it's actually kind of interesting. Okay, who invented the p-value? Where did this come from? Well, it turns out the concept of p-value was first articulated by a very famous British statistician and geneticist. He's known as the father of modern genetics, named Ronald Fisher. Okay, and he first kind of came up with the concept of the p-value in his very famous lady tasting tea experiment that he talked about in his book Design of Experiments in 1935. Is anyone familiar with the lady tasting tea experiment? Okay. Well, then this is actually be a lot of fun. Now, my first question is, how many of you drink tea? Does anyone here drink tea with milk? Okay, I don't drink tea with milk, so okay. My very important question to you, do you put the milk in first or do you put the tea in first? Which one do you do? What's your order? Oh, that's not helpful at all. What do you milk first? Okay. I don't drink tea with milk, so I don't know, but I do know that there can be a debate about which is the appropriate way, correct? And so, milk first, tea first. Well, what am I talking about? Well, Fisher was a scientist, and so one day he was having tea with a female colleague of his. This is actually, I found out, a true story. And he served her tea with the milk put in first, and she refused it because she said she prefers tea first, and she can tell the difference. Okay? So Fisher, being kind of the skeptical guy he is, and a scientist, said, I'm going to prove that. I'm going to design an experiment to prove whether or not she can actually tell the difference if you put the milk in first or the tea first. Okay? So he developed the very first null hypothesis. This is the status quo that we're going to assume unless we have evidence to assume otherwise. And so the lady claims she could tell the difference. So the status quo is the lady has no ability to distinguish between milk first and tea first. Okay? So what was the experiment he did? He prepared 8 cups of tea. 4 of them he put the milk in first. 4 of them he put the tea in first. And then he put them in random order and gave them to this lady. And so she drank all 8 cups of tea, and then he asked her to pick out the ones that had milk first. Okay, so essentially she had to discriminate between milk first and tea first. This was done all randomly. She obviously didn't know. All right. So the idea would be that can we find strong enough evidence from this experiment to either reject this null hypothesis or not? Okay, so what would you think? So obviously if the lady picked 0 cups correctly, we would know we would not reject the null. What about 1? 2? Probably not. That's like 50-50, right? But 3? If she picked 3 of the 4 cups correctly, how about 4? Maybe. All right. So I'm guessing you're probably all dying to know what happened. As it turns out, the lady picked all 4 cups correctly. She picked out the 4 cups out of 8 blindly that had the milk first. Okay. So the question is, how likely would it be if she had no knowledge and was just randomly guessing that she could have done that? And it turns out if you work out the math that the probability that the lady could pick all 4 cups correctly, assuming she had no ability to discriminate and was just picking randomly, is only 1.4%. Okay, very unlikely. Based on that, what did Fisher do? He rejected that null hypothesis and concluded that the lady was actually correct. She could discriminate a bit between milk first and tea first. Okay. This is the very first example of a randomized experiment. It's weird because we think of randomization taken for granted, but back before the 30s, people weren't doing this. This is also the first example of the idea of a p-value or using a probability to evaluate a null hypothesis. Okay, so based on this experiment, he devised this concept of a probability or p-value as a kind of an informal index, a sliding scale of evidence to measure the discrepancy between what you are hypothesizing, your null hypothesis, and what you observe. The greater the discrepancy between your hypothesis and the data you observe, the less likely the hypothesis is true. Okay? Does that make sense to everybody? P-values are kind of, listen in general terms. All right, so that's where the p-value came from. Now, this is in the 30s, and there were some definite critics of this system of thinking. In particular, there were 2 other statisticians named Lehman and Pearson. For those of you who've taken other stats classes, these names might seem familiar too. They criticized Fisher's kind of theory of this for a couple reasons. One, they said you only have a null hypothesis. If you notice that only was the null, there was no alternative hypothesis, and they felt that if you have one hypothesis, you should have an idea of what you would expect otherwise. The other thing they really disliked was this kind of idea of a sliding scale of evidence. They felt that was too subjective. So they devised a completely different system of doing experiments where the whole point was to choose between 2 competing theories to explain a phenomenon. And as long as that criteria met a certain threshold, we would reject what was now being called the null for the alternative, vice versa. So instead of this idea of sliding scale of evidence, it was really fit in a box, yes or no. Okay? From Neyman and Pearson, we get the idea of the null versus alternative. We also get the ideas of type 1, type 2 error, because the criteria are fixed above and before you run the experiment, and also power. Okay? Now, these two theories are not actually compatible. In fact, if you read letters back and forth, it's kind of funny because they kind of hated each other. They were always trying to put the other theory down. So it's kind of ironic that over time, what we have done is taken the idea of a Fisher p-value, where the smaller the p-value, the stronger the evidence against the null, combined it with this idea of a fixed criteria to choose one over the other, and this is currently what we do. And it really doesn't make a lot of sense if you think about it. It's like taking a duck and a horse, okay? We can argue about which animal is better, right? But we don't want to combine them into one. But this is essentially what we're doing. And because of that, because we're using a Fisher p-value in a way that was never meant to be, I'm going to submit that they are being widely misused and misrepresented in research. So first of all, 5%. Why do we use 5%? I mean, we're all taught 5%, but where does that come from? Okay, but when I first started learning about statistics, I was expecting there to be some really strong theory reason why 5% is this rigorous threshold, but it's not. Fisher's kind of suggested that back in 1935 as a kind of a level for convenience sake, but he never wanted to have a fixed cut point. He was more about this, again, this sliding scale of evidence. And you think about it, that really makes sense because p-values are probabilities. So if I told you that you had a 4.9% chance of being struck by lightning, and you had a 5.1% chance of being struck by lightning, we'd probably conclude that your chances of being struck by lightning are pretty similar. But the way we treat p-values now, totally opposite conclusions. If your p-value is 0.049, you say, oh, it's less than 0.05, reject. But if it's 0.051, you're like, oh, it's not less than 0.05, we can't reject, okay? Really, since p-values are probabilities, that doesn't really make a lot of sense to me. And because of that, and the reason also is that we have an over-reliance on p-values to make decisions. A lot of times when working with investigators, the first thing, I write up this long report, the first thing they do is they look at the p-value before looking at anything else. They want that p-value alone to drive decisions. But you have to remember that p-values are functions of sample size. Okay? If you have working on projects with really large samples, it's very easy to get small p-values by the way that we do them. Conversely, if you have very small sample sizes, small p-values are harder to get. And so again, really what this boils down to is that we need to make a distinction between clinical versus statistical significance. Okay, so here's a silly example. This was a Nature article that was published in 1998, which showed that males born in spring were taller than males born the rest of the year, which is kind of like, oh, that's interesting. Okay, this actually made the news, made it into Yahoo Health, and then was published elsewhere. And then you look at the actual article itself, and you see that the height advantage for males born in spring was 0.6 centimeters. Okay, now I have no medical training at all, but I'm going to say as a layperson, I'm guessing that's not clinically very meaningful, is it? So, but why did they report this? Because it was statistically significant. And if you ask why, look at the sample size. Using a large human male population of 507,000 males. So because of that large sample size, we were able to find statistically significant differences of something that probably is not clinically very meaningful, okay? All right, and then the last thing, so there's worries about p-values for that, but one of the biggest issues today we have with p-values is the idea of multiple comparisons, or fishing for significance, or p-hacking. And what do I mean by that? What I mean is running multiple tests, doing multiple subgroup analyses, slicing and dicing your data, and then cherry picking the p-values less than 0.05 and only reporting those, okay? That's what I call you're kind of going on a fishing trip. We know we want small p-values and people deliver them because a review article showed that almost every single article published has a significant p-value less than 0.05. And what's really interesting is just last week, did you see the news about the famous researcher from Cornell? He ran the Cornell Food and Branding Lab. He resigned after having 13 of his articles retracted, including 6 from JAMA, for bad statistical practices and just in general. And one of the things that his lab was notorious for was p-hacking. Okay, so this is definitely a problem. Just because you make it to be a very famous and well-respected researcher doesn't mean you're immune from this problem. I find that when we talk about it, so what does this mean? It means that when you do multiple tests, the chances of a false positive, getting a small p-value, particularly by chance, go up. And as a statistician, I can talk about this, but it's always hard to explain for researchers to understand. So because of that, and because you guys have been working so hard trying to pay attention and stay awake, I'm going to show you how this works with a game. So we're going to have a little fun now. I have a game I want to play with you guys. You're like a statistics game, oh my goodness. But I'm going to need 2 volunteers. And so because I know that no one ever wants to volunteer, I'm going to promise you a few things. First of all, this game will ask no hard questions or any questions at all. So you're not going to be embarrassed except for being in proximity with me. You will have very little to do, and I have prizes. Okay, so what I did is, and we want to have some fun now because we've been talking about statistics. Statistics is boring. I need a volunteer. Can someone please volunteer for me? Who would like to be a fun volunteer to play a really, really fun game? You volunteer. Yes, you, please. Okay, it's a game. Come on down. Here, let me get this. Come on down. Yeah, everyone clap. Yes, it's a game. Yay! Don't you love my high-tech game? All right, thanks so much. Thanks so much. Okay, that was enough. That's enough. Come on down. All right, great. So what's your name, please?
Julia.
Julia, nice to meet you.
Gardenia.
Nice to meet you. All right, I'm going to need you two to help me play a very simple stats game. So, Julia, come over here. I want you to play game number 1 with me. So the first game we're going to play, it's a die game. So we're going to roll a die 1 time, okay? If you roll a 6, you lose. Other than that, you win an amazing prize. Does that sound like a good game? You just have to do one thing. All right. Now, because it's hard to, actually have a die, but it's actually hard to see, I am going to use technology, if everyone's okay with that. It's a random dice roller, okay? And I'm going to let you roll 1 die, okay? So all you have to do to play this game is push a button.
Okay.
So can you go to the roll dice? Okay, everyone, let's root for Julia. Just press the button. Okay, ready? Not 6, not 6, not 6, not 6. Oh, no! You just got so unlucky. I am so sorry. Let me see if I can. I am so sorry, Julia. That was just unlucky.
Yeah.
I am sorry. But you know what? You were such a good sport that I'm going to give you the prize anyway. So here you go. A giant chocolate bar. Now you guys are wishing that you had done this, right? All right, thank you so much, Julia. Everyone, Julia, a round of applause. Julia was unlucky, don't you think? Okay, now, we're going to play the second game. I need to present this back here. Okay, so we already played this game. You're going to play a different game. You're going to roll a die 10 times. And if you roll a 6 anytime, you lose, otherwise you win. Okay, do you understand that game? All right, now what we're gonna do here is, because, I'm just gonna escape out of here. Again, because if you're okay with me, you can choose technology. So if you're okay with not rolling 1 die 10 times, we can just roll, if everyone's okay with that, we're just gonna roll 10 dice at once. Otherwise, if you guys have real strong objections, she can have her push it 10 times, but we're okay with her pushing it once, right? All right, okay, you ready? Yes. We're gonna roll 10 dice now. All right, everyone root for her. Come on, let's see what happens. Oh, just do it once, just once. Oh my goodness, you happen to roll a 6. I am so sorry. Hang on, we have to play this again. I am so sorry. But you know what, again, because you were such a good sport too, we're going to play. All right, everyone give her a hand. All right. Wasn't that an amazing game, guys? All right, now, as it always happens, as randomness happens, both of the contestants lost this game. But I'm gonna ask you a third question. If you had to pick one game to play, game 1 or game 2, which one would you want to play if you want to win? Game 1. Why? Right. But so the interesting thing about, the only difference between game 1 and game 2 is the frequency which you roll, right? If we're assuming it's a fair die, that means any single time that we roll a die, the probability of losing is only 1/6. unfortunately, unfortunately it didn't work. Odds were not in your favor that time. I always want to fix these examples so they work out exactly the way I want them. But instinctively, you are correct, because you know that the probability of rolling a 6 in any one roll, so game 1, the probability of losing is only 17%. But we understand intuitively that if we keep on rolling that die, the probability of getting a 6 at least once goes up. It's not 1/6, it's almost guaranteed, which is why I did it. I sort of gamed it. Same thing when we do testing. The same principle of a die roll goes to testing. So the way we construct our tests, we set what we call the type 1 error to be 5%. What does that mean? That means we accept in the long run about 5% of those small p-values, less than 0.05, are going to be false positives. Okay, we just accept that as, in research, that some of the small p-values we get are going to be false positives. And we set that probability to be low. But when you start multiple testing, just like rolling a die, that probability changes. So this is what happens to the probability of a type 1 error the more tests you run. So with one test, it's 5. With 10 independent tests, that probability goes up to 40%. And then with 50 tests, you are almost guaranteed to have a false positive, which is why, especially for those of you who work in high-dimensional data, for screening hundreds or thousands of genes, the problem of false positives in multiple testing is a huge issue. Okay, did that dice game help you at all understand that? At least I gave you some candy, so. Okay. All right. Because of p-values, because of the controversy over the misuse of p-values, in 2015, did you know a psychology journal actually banned p-values? So basically, they said they were not accepting any submissions that used a p-value, which, again, probably you did not notice that, but in the stats world, this was huge. This was just like all people were talking about, because it was just such a dramatic shift. There was so much confusion that the American Statistical Association took the unprecedented step of getting together some of the top statisticians and releasing a joint statement on the use of p-values in research. And this is still an active area of debate. In fact, last year I went to this symposium where we actually were talking about p-values. I mean, imagine that, a conference of statisticians, a multi-day conference of statisticians, and we're talking about p-values. I mean, just imagine that. It was amazing. All right. So what does this mean for you? Practically, what does all of this what are the implications for you? Okay, well, I don't think, honestly, that in the short term, p-values are going anywhere in research. There's a growing awareness that we need to be careful with them, but honestly, p-values are still being used, and you're going to still be dealing with them. What are my practical recommendations for you? Number 1, p-values should never be used alone to determine whatever you call significance. Keep in mind there's between clinical versus statistical significance. When you're reporting results, never just give the p-value. Give an effect size. Tell how different those groups were. Give an idea of the variability. Don't just say p less than 0.05. Also, most important, we need to be better about being transparent in how we report results. No cherry picking of results, okay? If you screened 1,000 genes, we need to say that in the paper. We don't just pick out the one gene that showed differential expression. And the third thing is we need to avoid p-hacking by, again, focusing our research, having better designs, not letting the data necessarily drive our hypotheses, but really trying to come up with a focused plan of research. And again, that's where a statistician can help you out. So these are my practical recommendations for you. Just be careful of p-values, okay? We're almost out of time here, so very quickly, my last piece of advice to you. My first one was to talk with a statistician before the data are collected. The second one was to be careful of p-values. The third piece of advice is this really does matter. So I know a lot of times I'll talk, and as a statistician, I'll talk, and you might say, yeah, I agree, but you know what? I have a small project. It's a pain to work with you guys. It's so much easier. I can just do this. I just need to get this little paper out. Does it really matter? And I'm going to say it really does matter. You might want to say don't sweat the small stuff, but like in everything else, it's all about the details. And let me show you why I think it really matters. No matter how small the project, we need to be very, very vigilant about giving the best science we can. Here's a paper that was published in Science in 2005. Okay? It's called visfatin, a protein secreted by visceral fat that mimics the effects of insulin. People were very excited when this came out because they were thinking about all the therapeutic benefits and experimental benefits. Well, this paper happened to be retracted 2 years later due to issues that were discovered. To be fair, the authors protested it, but the journal and the advisors told them to withdraw this article. But the problem is that this article, even though it's been retracted, has not died. So there's a great site called Retraction Watch, which kind of follows papers that have been retracted. I want you to look at this. At the number of citations this paper had before it was retracted. In the 2 years it was published, it was cited 276 times, so it was pretty well cited. Look how many times it's been cited after this paper has been retracted. I actually just pulled this last week. I had updated this from a year ago, and there's been another 200-something citations just from last year to update this. And what bothers me the most is when you look at those citations, not all of them appear to be aware of the fact that this paper was retracted. So here's an example from 2016, which cited this paper. And if you look at the way they talk about it, no apparent awareness that this paper has been retracted. Okay? And then I went further and I found an even more current example. This one was published just in June of this year. And it still talks about, references this retracted paper. So my challenge to you and just my encouragement to you is that it really doesn't matter, no matter how small your research project you might feel it is, or even how large. If you are publishing something and getting it out for public, essentially it's out there forever. So bad science is bad science. So in conclusion, statistics is just so much more than just pushing buttons and getting p-values. There are so many facets to kind of combat the idea of bad statistics in research. And bad statistics are a major culprit in the problem of irreproducible research. So we have the design, the analysis, and the interpretation all involve statistics. All three of these actually have to be correct for your research to be correct. If any one of these are wrong, then your research is going to be flawed. And definitely, statisticians, we really want to be involved as collaborators with researchers. So we want to work with you from the very beginning. We don't want just to have the spreadsheet with column A, column B. We really want to work with you to understand the science behind what you're doing. We want to be partners to help you make your research as strong as possible. So of course now you're all excited to talk with a statistician. Sometimes I do even have candy, so if you want to work with me. So feel free, interested in talking with a statistician. My name is Amy Lehman. I work at the Center for Biostatistics. Our website is biostatistics.osu.edu. My colleague Guy Brock will be talking later this afternoon about reproducibility and rigor in research. So hopefully I just kind of brushed on some topics that he'll be going into more detail later. So thank you so much for your time. Thank you. Any questions?
What was the name of that website where you can identify all the articles that are written?
Oh, it's called retractionwatch.com. It's a kind of a depressing website. They're kind of a watchdog website that Retraction Watch. So if you just Google Retraction Watch, that will do it. They follow lots of different retractions for reasons all the way from fraud to just bad, irreproducible data, something like that. Yep. Any other questions? Okay. All right. Thank you so much for your time. Appreciate it.